Dear students, I would like to continue the lecture about the ear. In the first lecture, I told you the parts of the external ear. I showed you the auricle, the external acoustic meatus, and the eardrum. And also, I demonstrated for you the middle ear with the tympanic cavity, the auditory tube, and the auditory ossicles. And today, I would like to show you the parts of the internal ear, the bony and the membranous labyrinth. Here you can see that preparation, what I will show you in the demonstrative part. The higher magnification of the inner ear you can see in this picture. It has two main parts. The first part, which is for the hearing, is called the cochlea, and the other parts, the vestibule and the semicircular canals. These are the parts of the vestibular system. Here you can see the nerve, the vestibulocochlear nerve, which will collect the information from the receptors of these organs. If you see the membranous part, which is located within the bony labyrinth. So here you can see the bony part. This is the membranous part in this picture. We can distinguish two main spaces between them. Between the bony part and the membranous labyrinth, we can find a fluid which is called the perilymph, and that which is located within the membranous labyrinth is called the endolymph. But what are the parts of the membranous labyrinth? I would like to start first with this, which is located within the cochlea. Within the cochlea, you can see a tube which is called a duct, which is called the cochlear duct. It's a blind duct here you can see with 2.5 turns, which will go around within the spiral canal of the cochlea. After the next part, here if we see the vestibule, within the vestibule we have two main membranous parts. The first is the saccule, and the other is the utricle. Of course, between them we have connections. And finally, within the semicircular canals, the bony part, we have the same semicircular ducts which will form connection with the utricle. Here you can see another part which is called the lymphatic duct, which is the protrusion of the membranous labyrinth, and here are the connections between the utricle and saccule, the utriculo-saccular duct, and between the saccule and the duct, the cochlear duct, is called the uniting duct. First, I would like to talk about the semicircular canals. I would like to show you the function of them and the position of these structures. We can distinguish three different semicircular canals. They are located in three different directions, because the function of them is to fill the angular acceleration of the body of the head in different dimensions. So here you can see the three different semicircular canals. So what's that? The first one, which is located posteriorly, this is the posterior semicircular canal. The axis of this will be parallel with the axis of the petrosal part of the temporal bone. This is the posterior semicircular canal, and of course the duct, like the part of the membranous labyrinth. After the next semicircular canal, it has perpendicular direction. It is perpendicular to the axis of the petrosal part of the temporal bone. This will be the anterior semicircular canal and semicircular duct. And finally, laterally you can see the lateral semicircular duct and canal, which has which is perpendicular to the first two ones, and it is located all in the horizontal direction. This is called the lateral semicircular canal and duct. If we see a picture from the internal base of the skull, here you can see the scala media, the fossa scali media. This is the posterior cranial fossa, and this is the petrosal part of the temporal bone. Here we can see very well the cochlea and the semicircular ducts and canals. The posterior one will be parallel to the axis of the petrosal part, and you see the axis of the petrosal part will form 45 degrees with the mid-sagittal section, and the anterior semicircular canal will be perpendicular to the axis and 45 degrees with the mid-sagittal plane. And this is the lateral semicircular canal, and it's three-dimensional. So within this picture you can see very well the directions, the positions of the semicircular ducts and canals. If we see one semicircular duct, one semicircular canal, it has two parts, and you see two ends. The one is bigger, is dilated, and we have a simple crus there, posterior. So this is the crus simplex, and anterior, here you can see the other part will form a dilatation. It's called the ampulla, crus ossum ampullare, ampullary part. Here we can see the receptor of the semicircular canal, which is called a crista. It is responsible for the angular acceleration of the head. You can see that we have three semicircular ducts, but the two semicircular duct, two simple part of the semicircular duct will be fused, and we have a common crus, the common crus of the anterior and the posterior semicircular canal. So we have 6 semicircular ducts, but we have only just 5, openings to the utricle, because of that here the two simplex, the parts of the semicircular canal, they are fused there. Let's see the receptors of the vestibular system, and let's see first the receptors of the sacral and the utricle, the two parts which are responsible for the linear acceleration. Here you can see we have two receptors. The receptors, they are called macula. These are the maculae. We have one in the horizontal direction, for the horizontal acceleration, and one we have in the vertical direction for the vertical acceleration, and we are traveling with the elevator. So if we see the microscopic structure of this macula, I would like to show you the nerves, of course, which will collect the information from the receptors. So the utricular nerve from the utricle and the sacral nerve from the sacral. And here you can see the microscopic structure of the macula, which consists of special sensory cells. Here you can see the superficial modifications of the sensory cells, which are stereocilia, and one kind of cilium we have there in one side. And all of them, they are, first we have supporting cell, and all of them, they are embedded in a jelly-like material, which is called the autolytic membrane. And within this autolytic membrane, there are a lot of crystals, autolytic crystals, which are embedded within this jelly-like material. They can help, to move this jelly structure in horizontal and vertical direction. So these are the autolytic crystals on the surface on this jelly-like material. After the other receptor of the vestibular system, these are the ampullary crests. These are located within the ampullary parts of the semicircular canals, the semicircular ducts. This is, here you can see the ampulla of the anterior semicircular duct. This is the ampulla of the lateral semicircular part, and this is the ampulla of the posterior semicircular canal, semicircular duct. And you can see that all of them, they have an individual, an own nerve which will carry the information from the receptors. If you see the higher magnification of the receptor, which is located there in the ampulla, it has a special semi-lunar shape, like the crest, and if you see the cross-section of this crest, it has a special shape like a mushroom. And these are the neuroepithelia of special sensory cells and supportive cells, similarly to the macula. These are the superficial modification of the cells which are embedded in this jelly-like material, which is called a cupola, like a cap on the top of this crest. And as you see, we have no crystals there because the activation of these receptors is based on the passive movement of the endolymph, which is there within the semicircular canal. So when we move our head like this, the fluid which is located within the semicircular duct, it will move this cupola, on two sides, and this will be the activation of the cells which are located there within the crest. Let's see the activation of the cells. So the stereocilia, they are located here. Here we have one kinocinin there on the top. And if they go close to each other or towards each other, this will be the activation. It means the activation of the cells. So when we are traveling with car and we do longitudinal or linear acceleration, in this case, this jelly-like material with the autolytic crystals will move and will activate the cells. And the other, the activation of the crest. So if we move our head like this, the endolymph will move within the semicircular duct and the semicircular canal. We will move this uppermost jelly-like part of which is called the cupola, and this will be the activation of the cells here in the crest. And finally, I would like to show you the histological picture of the macula and the crest. Here you can see the cells and the supporting cells, the jelly-like material, this autolytic membrane you can see on the top, and these are the autolytic crystals. And this is the cross-section of the crest, which is normally in like a mushroom, but maybe you could have also in another section which could be similar to the macula, just for example, if you see only just one part of the crest, it's so similar to the macula. But what's the difference between the macula and the cresta if you have only just this part of it? In the macula, you can see the autolytic crystals, which are located on the surface of the autolytic membrane. In the second part of the lecture, I would like to show you the structure of the cochlea, which is responsible for hearing. The cochlea, here you can see the cross-section of the cochlea. The lower part is called the basis of the cochlea, and the apical part is called the cupola of the cochlea. And there, around, we can see the semicircular canals, and the center, which is called the modiolus, will form the axis of this whole structure. Within the modiolus, we can see longitudinal canals. Within these longitudinal canals will be the fibers which will form the cochlear nerve, which will originate from the spiral ganglion, which is located there in the peripheral part of the modiolus. So around the modulus we can see a spiral canal, spinal also canal of the cochlea, which will form 2.5 turns around the modulus. And there, in the middle part, there you can see a spiral lamina. And the spiral lamina, this will be the end of the spiral lamina there in the cupola, which is called helicotrema, and the end of the spiral lamina is called the hanulus of the spiral lamina. If you see the position of the membranous labyrinth within the cochlea, you know, the cochlear duct is the part of the membranous labyrinth which is located within the cochlea. The cochlear duct is located there in the middle part of the spiral canal. That's why we can say a scala media, a middle scala, and it's fixed to the lateral wall, and it will divide the cavity into 3 different parts. So the middle one is a scala media, which is formed by the cochlear duct. This is what I mentioned. The upper part is called the scala vestibuli. The scala vestibuli will form connection with the vestibular window where the base of a stapes is fixed. So this how the stapes will move here the endolymph, you know, which is located, the perilymph, which is located there, you know, endolymph we have here in the cochlear duct, and also there we can see the other part, which is called the scala tympani. You see the scala vestibule is going up, and there we'll turn back and we'll continue to the scala tympani there, and the scala tympani will finish at the cochlear window where we have the secondary tympanic membrane, which will withdraw the unuseful waves of the perilymph. So here we can see the 3 main parts: the scala vestibuli, the scala media, and the scala tympani. Here in the spiral lamina, there in the proximal part of the spiral lamina, there we can see the spiral ganglion. These are bipolar neurons. The peripheral process of these neurons will come from the receptor cells, and the central part will form the cochlear nerve, which is located in the longitudinal canals of the modulus. Let's see now the higher magnification and the histological picture of the spiral canal of the cochlea. So here in the middle part, this is the spiral lamina, spiral also lamina there. Into this part is fixed the limbus of the spiral lamina. It has 2 lips. The upper part is called the vestibular lip, and the lower part is called the tympanic lip. From this will originate a membrane which will divide, which will separate the scala vestibuli from the scala media. This is called the vestibular membrane or Reissner membrane, and the other membrane is located between the scala media and the scala tympani. It's called the basilar membrane. On this basilar membrane will sit the receptor, the cortis organ, which is responsible for the hearing. What are the other structures what we have to mention there? The basilar membrane is fixed to the lateral wall by the spiral ligament, and there we can see stratified epithelium, which is called the stria vascularis. It is responsible for the production of the endolymph. So we know that here we have endolymph, and there in the vestibular and the tympanic scala, there we have perilymph. And there on the basilar membrane we can see the cortis organ. And now I would like to show you the structure and the cells of this special part of the cochlea. So First, if you see the higher magnification of the cells which are sitting on the basilar membrane, first we can distinguish two special cells which will form a triangular shaped tunnel, cortis tunnel in the middle. These are called the pillar cells. We have one inner and one outer pillar cell. Here is the modiolus in the middle, so that's why this is the inner part, this is the outer part of this organ. So inner pillar cell and outer pillar cell is visible there. And we can see there on the apical part, these are the hair cells. We have one row of inner hair cell and 3 rows of outer hair cells, and we have almost 1,000 cells within one row. But it is really important to know that the hair cells, they are not able to regenerate. So if unfortunately if we have a lesion, in this case that cell will die and we have no receptor in that part. Of course, it's no problem if you have only just one or some cell loss, but if, for example, we have a huge region where all of the cells they die, unfortunately, this frequency, we lose this frequency within the hearing. It is really interesting that there are some animals, for example, the birds next to the big waterfalls, though where we have a very big load, those cells they are able to regenerate, but in humans not. So that's why we have to take care of our hair cells, and it's not allowed to use a very loud, how you say, voice with the headsets. Okay, so the hair cells, inner and outer hair cells, and around the hair cells here you can see the phalangeal cells which support them. They have different author names, but for you it's not necessary to know all of these spotlight details and so on. So this picture, this three-dimensional scanning electron microscopic picture, they show you the position of the stereocilia, which are located on the top of the hair cells. They have a very special shape, V shape. Here you can see one row of the inner and 3 rows of the outer hair cells. And there you can see the processes of the phalangeal cells which are going there towards the neck portion of the hair cells, and they form a separated barrier there on the top. And that's why here this area is separated from the endolymph. So there we have another fluid which is called the cortilymph, is separated from this. So the stereocilia, they are located in the endolymph. And this is the histological picture, what you can see in the histo slides. This is the cross-section of the cochlea. This is the modiolus in the middle with the nerves, the cochlear nerve, and the cartilage or the ossification which will surround the cochlea is visible there. And we can divide the spiral canal into 3 main parts. In the middle, the scala media formed by the ductus cochlear duct, ductus cochlearis. The upper part is the scala vestibuli, and the lower part is called the scala tympani. And here, if you magnify this blue or purple part, this is what we can see in the slide. So the limbus, which is located medially. After the upper part is called the vestibular membrane, the Reissner membrane, which will be the border between the scala vestibuli and the cochlear duct. Here you can see the basilar membrane, which will be the border between the cochlear duct and the scala tympani. There the spiral ligament and the stereovascularis is visible. This is the limbus, and in the middle here you can see the cortis organ. And if I magnify the histological picture of the cortis organ, there you can see the limbus, and from the limbus will originate a jelly-like material which is called the tectorial membrane. And here you can see the cells In the middle, the pillar cells, the inner and the outer pillar cell will form this triangular shaped tunnel in the middle. There we have one hair cell, inner hair cell, and 3 outer hair cells. And under we can see the supporting cells. So those are the cells what we have to distinguish in the histoslide of the cortex organ. And how do they work? Behind the activation of these hair cells, we have a really complex physiological background, which was described by Békési. It was a Hungarian scientist, a physiologist, who described and got a Nobel Prize at the beginning of 1961, a Nobel Prize, because of the description of this mechanism. How it's working. So here you can see that through the external auditory canal will come, the air waves like this, which will move here the eardrum, and this will move the bones of, the malleus, incus, and the stapes. The base of the stapes will move in and out in the window, and this will move the fluid which is located there, which is the perilymph there in the scala vestibuli. These waves, they are going up towards the cupola of the cochlea, and after will in the helicotrema will turn back to the scala tympani, and there the waves, they are absorbed within the secondary tympanic membrane. And these waves, they will move the basilar membrane there, and where we have, depending on the frequency, the different part of the basilar membrane will form the biggest amplitude. And this is what it means that we will hear the lower voices in the apical part of the cochlea and the higher voices in the deeper part of the cochlea. So here the vestibule, the scala tympani, this is what I told you. And you know how the basilar membrane will move. Here you can see the stereocilia of the hair cells. They will form connection with the tectorial membrane, which is located there on the top. So where we have the bigger amplitude, there we have the bigger activation. So this was the mechanism what György Békési described for the activation of the hair cells. But Dr. Todt will talk about all of this mechanism in detail in the next lecture. Here is only just a private picture with Professor Leglödi and Lubitsch. We had a very good tour in Innsbruck, and there was a very special museum. Here you can see the hair cells. All of the hair cells represent one frequency, and with the movement, you see here we are moving these special processes. We could play one song. It was just funny, I wanted to show you this private picture. And finally, I would like to tell you some words about the development of the ear. Here you can see the schematic picture from the 22nd day of the embryo development. This is the rhombencephalon here, and this is the ectoderm, and on the top of the ectoderm the otic placode is formed. And after here you can see the invagination of this otic placode to the deeper part, and it's called otic pit. And finally it will form the vesicle when it will lose the connection with the surface. And next to the otic vesicle will be the vestibulocochlear ganglion, or the ganglions which are originated from the neurocrest. So from this otic vesicle will develop the membranous part of the inner ear. We can divide it into 2 main parts. There you can see a ventral part. From the ventral part of this will develop the saccule and the cochlear duct. And from the dorsal part will originate the utricle, from the utricle the semicircular ducts, and the endolymphatic duct. So these are the main parts which will develop from this vesicle. If you see the development of the cochlear duct from the ventral part, the cochlear duct first is just a small blind protrusion of the ventral part of the otic vesicle, but after you see it is elongated and will form 2.5 turn to form the whole cochlear duct, which will be within the cochlea. And this, the connection between the sacral, will be the ductus reuniens or uniting duct. This cochlear duct is located in the cartilaginous shell, and here you can see the cochlear duct. This is the cartilaginous shell around, and there it will vacuolize to form two parts, the scala vestibuli here, and the lower part will be the scala tympani, and here you can see the cochlear duct. First the cochlear duct, the cells they will be similar, but after they will differentiate and they will form an outer ridge and an inner ridge. From the outer ridge will be the cells, the neuroepithelial cells and the supporting cells of the cortex organ, and from the inner ridge will be the cells of the limbus. And on the top you can see the development of the tectorial membrane. So what are the cells which will be from the outer ridge? The outer hair cells, the inner hair cells, and the pharyngeal cells. Here you can see the pillar cells, and from the inner ridge will be the limbus. And this is the the nerves, the auditory nerve fibers will bring the information from the hair cells. This is what I told you. And the dorsal part from this will form the utricle and the semicircular duct, semicircular canals. Here you can see that first from the utricle we have just a flattened out flattened protrusion, but it will communicate with the cavity of the utricle first, but after you can see that the walls, they will fuse and this middle part will disappear. So finally we have just this semicircular shape, semicircular ducts. And the semicircular ducts, they have dilated part is called the ampullary part, and the other is called the crus simplex. But we know that the anterior and the posterior semicircular canals, they are fused, so that's why they have a common crus simplex. And here within the crest, here within the ampulla, we can see the crista, the crest, the receptor, and within the utricle and sacral, there we have the maculae, the receptors of the linear acceleration. The crest, they are for the angular acceleration. Here you can see the development of the external acoustic meatus and the tympanic cavity with the Eustachian tube. So you see that the external acoustic meatus is developing from the first pharyngeal cleft. So there we have ectoderm, and the eardrum, the outermost part of the eardrum, is originated from the ectoderm, while the tympanic cavity with the tubo-tympanic recess will originate from the first pharyngeal pouch. So there we have an endoderm, and between them we have a little connective tissue. And you can see that parallelly with these structures will develop the bones too, which are located in the tympanic cavity. The bones, they are originated from the pharyngeal arches. So here you can see the first pharyngeal arch will form Here you can see the cartilage of the first pharyngeal arch, which will be the base of the mandible. And from the first pharyngeal arch will be the malleus and the incus. This is the ligament, the sphenomandibular ligament, which is located between them. And we know that that muscle, which is the tensor tympani muscle, which will be attached to the malleus, this is innervated by the trigeminal nerve, because we know very well that the nerve of the first pharyngeal arch is the trigeminal nerve. And after there you can see the second pharyngeal arch. From the second pharyngeal arch will originate the stapes, the sapes, and here you can see the styloid process and the stylohyoid ligament with the upper part of the hyoid bone. And we know that the muscle which will be attached to the stapes, it is the stapedius muscle, and the innervation will be by the facial nerve, because we know very well the nerve of the second pharyngeal arch, this is the facial nerve. And finally, here you can see the development of the auricle. From the first and from the second pharyngeal arches, you see we have three auricular hillocks, which will fuse and form the final shape of the ear with the helix and the antihelix. As you see, it's a really complicated developmental step, so that's why we could have developmental malformations, of course. And it is always really important sign if you can see a newborn with developmental malformation of the ear, because it can show you other developmental malformation too, because we know that the development of the pharyngeal arches it has connection with the development of the heart too. It's combined with heart developmental malformations, because the neurocrest cells, they are involved in the development of the heart too. So if you see a newborn with malformations in the ear, always we have to examine for the further development of genetical malformations. And of course, other environmental effects, they can cause also deafness, not only just genetical, some inflammation too. So I started with the ear, how important is the shape of the ear, and I finished my lecture with this too. And I would like to thank you for your attention.